Hi, everyone. My name is Connor Kubo. I'm a graduate student here in Genome Sciences, co-advised by both Nobu Hamazaki and Jay Shinduri. And today I'll be telling you about my project, which is reading the genetic instructions on making the human body plan. So to start off, as humans, we all start from a single cell. And through cell division, the cell is able to replicate and form a cell mass called a blastocyst. However, this blastocyst, the cells that make up the embryo, consist of a single cell type and don't have much patterning. And in the third week of development, they undergo a process called gastrulation. And gastrulation is this complex process that gives rise to all the cell types that make up the human body, as well as establishing the human body plan. So gastrulation is largely important for development, and when things go wrong, you could imagine that developmental disorders or potentially loss of the embryo could occur. Now, the problem is human gastrulation is very difficult to study because embryos at this stage are extremely hard to come by. And it suffers from this sort of Goldilocks paradox, where samples from in vitro fertilization are too early to study gastrulation, and then samples from fetal samples are often too late for gastrulation. So to study this time point, people in my lab, Nobu and Wei, have developed this stem cell derived process to make gastruloids. So these gastruloids are these stem cell derived embryoid models, and they largely recapitulate the morphology that we would expect in a human embryo at a certain stage, Carnegie stage 11. And when we do single cell RNA sequencing on it, which is a method for getting the gene expression in an individual cell and then plotting them based on this gene expression, we can see distinct cell types that will give rise to cells later on in human development. And we see an endodermal lineage, which gives rise to the lining in the digestive tract. We see a neural lineage, which gives rise to the early spinal cord, as well as neurons. A cardiac lineage giving rise to muscle in the heart, and a somatic lineage, which gives rise to muscles, skeletal muscle, bone, and connective tissue. Now, I'm interested in learning how the genome encodes for each of these cell types. So to do so, I want to create these gastruloids, but perturb specific genes in them, and then read out the changes in the cell types to find out if these genes are important for the development of that. So to do so, first, I knock out genes of interest using CRISPR in human embryonic stem cells. I then generate gastruloids from these knockout cell lines. And then finally, I phenotype them using single cell RNA sequencing. And this is an example of single cell RNA sequencing data from the last time point in the embryo. As you can see, there still are 5 different lineages there. All right, and next I'm going to show you a couple interesting stories from genes that I've knocked out. So first, we have a proof of concept. So this is hand1. This is a gene that's important. Well, it's well known that it's important for the establishment of cardiac development. And mutations in hand1 have been associated with congenital heart disease. So when we knock out hand1, we would naturally expect to see development of cardiac muscle to be perturbed. And this is the single cell RNA sequencing data in our hand1 knockout gastruloids. So as you can see, we have the presence of the 4 different lineages. However, for our cardiac cells, you can see a depletion in them. And this is what we would expect. Next, so that was just a proof of concept, but next I looked at the knockout of a receptor called GPR161. And this is a receptor that's involved in sonic hedgehog signaling. In humans, it's known to play a role in breast cancer development. And in mice, it's known to play a role in neural tube development in developing embryos. However, not much is known about its role in human development. So this knockout we thought would be particularly interesting to look at. And when we knock it out, we actually see a novel cell type. We see depletions in all 5 of our different lineages: the cardiac, renal, somatic, neural, and the other lineage. And then we see the presence of a brand new cell type here in purple, which I have with a question mark. And since we have single cell RNA sequencing data, we are able to dive into these cells and figure out kind of what the cell type is. And right now we're still in the process of that, but later on we'll have a better idea. All right, so these are just 2 examples from the knockouts that I've done, but I've actually scaled up to 23 total knockouts. And we actually have significant phenotypes for almost half of them. So this is a lot of data to work with. And we're also planning on scaling up to 40 plus in the current, for our current progress. Now for future directions, I'd like to use this system to scale up to 200 plus genes. And this will be in collaboration with multiple different labs involved, or with funding from the Chan Zuckerberg Initiative, Allen Institute, and UW Labs. So by scaling up to 200 different genes, we'll be able to guide these clinical research trials and eventually get that to help them focus in to develop therapeutics or potential cures for these early genetic disorders. On top of this, we're also planning on comparing these knockouts with developing zebrafish and developing mice. And this will give us comparative knowledge on what's important to make a human versus what's important to make mammals or animals in general. So circling back to the title of the talk, at least, what I'm planning to do with this system is to understand how the human body plan develops and how this genome controls for it with the genes. And also maybe further applications could tell us about what makes us human. So if you're interested, feel free to reach out to me. My name is Connor Kubo again. I'm co-advised by Nobu Hamazaki and Jay Shinduri, and my email is connork3@uw.edu. And I'd like to thank my 2 advisors, Nobu Hamazaki, Jay Shinduri, as well as people from the lab, Wei Yang, Beth Martin, and Ridderman Garge, as well as other members of the lab. Thank you.
I think we could take 2 questions before we go on our break.
I want to ask that for the genes that you knock out for those embryos, what organism is that?
Oh yeah, so for the genes that we knock out in our system, it's a human stem cell derived system. So not really a true embryo, but more of like a model of a human embryo.
Oh, okay. Thank you. So there is like no ethical concerns about that.
Yes, that's a really great point. So I have a slide for this. Okay, so when comparing our gastroloid, which is our stem cell embryoid system, versus a human gastroloid or a human embryo, we're actually missing most of the anterior structures in our embryoid, meaning that there's no brain, so there's not capable of thought or anything, or developing past more than 5 days. So.
Okay, thank you.
Yeah.
One more question. We could do 2 more questions.
So for the plots showing the phenotype with single cell RNA sequencing, I was wondering what the X and Y axes represented.
That's also a good question. So to get the coordinates for these plots, we use a dimension reduction process called UMAP. It's pretty complex, and the actual XY coordinates on the UMAP plots are not super important, but more important is how close certain cells are to other ones on the plot. And that kind of gives an idea of similarity between certain types of cells. And that's why I'm able to kind of draw these circles around certain clusters. and call them certain cell types. Yeah.
I had a question related to the gastroloids. You said that they were created because of difficulty in the timing of human development, but you did mention that you were going to look into mice and the zebrafish. Would you produce gastroloids for those as well, or is it easier to work with them?
Yeah, so we had that thought too. For zebrafish, we'll do natural embryos because they're pretty easy to come by. For mice, it's a little more difficult, so we were thinking of doing mouse gastroloids. Yeah.
Thank you.